Well, children, the previous lecture we have in lecture 3 that we have discussed about the structure of the chromosomes, their arrangement and all. And today we are looking at the cell organelles and the structures involved in the cell division. Now, first thing is the nucleus. Nucleus, where the cell has the chromosomes, that is the main part of the cell division. Now, what is happening in nucleus? You see, this is a nucleus, the chromatin network is there, and you have nucleolus is there, and there you will find in animal cells the two dots, these are known as the centrioles, and this is known as centrosome. The centrosome and centriole act, they are the characteristics of the plant animal cells, and they are responsible and they act during the cell division. Well, so nucleus is having the nuclear membrane, where there are pores are there, is known as nuclear pore, and you have nuclear membrane is there and nucleoplasm is there. All right. Now we are coming to see what are the requirements are here. So centrosome and the microtubules are the things which are required for the cell division. Number one, the centrosome. Centrosomes are having centrioles. These centrioles, I mean to say these centrioles, the two dots, during the cell division, they just got polarized. They go towards the opposite ends and they make the right angle positions. So the centrioles placed at right angles, and this is duplicated during the S phase. They are duplicated during the S phase of cell division. Now you come, they grow out of the centrosome with, now this is the centriole, then we have the microtubules. The microtubules, they grow out of centrosome with ends growing towards the metaphase. So you know the phases change, prophase, metaphase, anaphase, telophase, like that there. You see, they grow out of the centrosome with ends growing towards the metaphase plate, and the microtubule spikes radiate and radiate from the central side occupied by centrosome, which is the primary microtubule organizing center, and in short which is known as MTOC. So primary microtubule organizing center. The cytoskeleton transport this, and the microtubule is thick, strong spirals of 1000 subunits. They are together 1000 subunits made of the proteins, and these proteins are known as tubulin, Tubulin, tubulin protein, they make them. Now the next organelle, I mean to say, the cell has the cytoplasm, and inside the cytoplasm there are miniature organs. You know, you have hands that works as one organ, leg that works as another organ, external organs. Then we have internal organs like heart, lung and stomach and all those, but they have a specific function. But inside the cell, the status of an organelle is exactly like that of an organ. They are specified miniature organs working for a definite and a particular purpose. So they are the organelles, and these organelles I told you as coming to this, the centrosome is also an organelle, and then we have a nucleus coming to the ribosome. Ribosome, as you know, is the seat of the synthesis of protein, that when we have studied the cell, we have come to know ribosome helps in the synthesis of protein. Right now, we need to know that what help does ribosome do during the cell division. Because in cell division, proteins are very much required, because they grow and even they create the new proteins, and lot of energy is required to just divide the cell. So that is what the ribosome is firstly doing. The ribosome, the place of protein synthesis, ribosome plus mRNA to form the polyribosomes or polysomes. Polyribosomes are also known as the polysomes, and cell will not copy DNA preparations for the cell division, for the cell division, unless there is if the cell is confirmed that we have lot of protein preparation system, the ribosome has all this capacity and all, then it will start copying the DNA. So that is what is said, the cell will not copy DNA preparation for cell division unless there is enough ribosome to make new proteins. I told you just now, new proteins. And this copying process happens during interphase, and this copying process happens during the interphase, because copying DNA requires so many proteins, I told you earlier also, and so much energy. Thus, number of ribosomes in a cell is a form of regulating cellular homeostasis. Well, children, the next organelle is the mitochondria, known as the powerhouse of the cell. It is known as the powerhouse of the cell. Here is the section of mitochondria, and you see the diagram, the matrix, formation of ATP is done here, ATP synthesis, and these are known as the cristae, and membrane of the space here, and then you see the ribosomes, dot structures, and granules are there. We have DNA formation is done over here, and this is the outer membrane and this is the inner membrane. Now, what is happening? You see, cells require lots of energy, the form of ATP, in order to synthesize the bioactive molecules. The bioactive molecules that will just divide the cells. So bioactive molecules are done in this way, bioactive molecules. Now, bioactive compounds are lipids, proteins and nucleotides for cell division. ATP production in mitochondria is also vital to maintain the other activities and to regulate the other activities, regulation of the other activities of the cell volume. Solid concentration and the cellular architecture. Cellular architecture means by how the cell is made, cellular architecture. And then they act as the driver of the cell division, because they supply the energy, then the cell divides. When they are satisfied that there are a lot of ribosomes in there and plenty of energy is there, then they divide. They allow the proper segregation of the organelles between the daughter cells of the cell division. And now the cell division. The cell division is a fundamental and active biological process by which a cell produces its own replica, having the similar structure, genetical and physiological properties, and brings upon the continuity of life. Now we have the significance of the cell division. What is the significance of cell division? It is prerequisite for the community of the life and forms the basic or evolution to the various life forms. If there is no cell division, no life will be continued on the surface of the earth. Unicellular organisms, they divide by certain kind of cell division, and this is known as asexual reproduction, because they divide directly into two. In sexual reproduction, we have the mitosis and meiosis, keeps continuous division of the cells, The multicellular organisms like the single cell is called, that starts from the zygote. What is zygote? A fertilized egg. The male unit, which is known as the sperm, and the female unit, which is known as the ovum, they get together and they are fertilized to form zygote. And this zygote in time slowly and gradually undergoes metamorphosis, slowly and gradually they divide and continuously divide to form the new organism. So that is what is called the characteristics of the higher organisms, and there the mitosis and meiosis both are there. Well, by successive divisions they do it, and the cell division is the basis of the repair and the regeneration of the old and worn out cells. We have a lot of activities going on inside the body, the metabolic activities, and for that there are a lot of cells which are damaged, and this could not have been repaired or amended if there was no cell division. So by cell division, that is repaired, and that is going, the system goes through. So this is how it changes. Now we have the types of cell division. We have three types of cell division commonly. They are known as the mitosis, that is the direct division. What I told you regarding the unicellular organisms like amoeba and all, they directly divide into two daughter nuclei, and that means two daughter cells. They are doing it by mitosis. That is the direct division. Second one is the mitosis. In mitosis, indirect equational cell division is there. Indirect equational cell division. And third one is the meiosis, indirect reductional cell division. Why it is called reductional cell division? Because here the number of chromosomes at first increases and then it reduces to half. So that is how it goes on. Now we should just discuss about the mitosis. Occurs in the prokaryotic organism. Prokaryotic means simple organisms, ancient and primitive type of organisms like protozoa. And you know amoeba is a protozoa, so in protozoa it is formed. Here, somatic cell is divisible into two parts, each grows to an identical organism that is formed. And the next is the mitosis. In mitosis, what is happened? It is a somatic cell division, body cell division it is known as, and it is happening in the eukaryotic cells. And single mass that goes under the daughter cells and divides into the daughter cells which are identical. The daughter cells must be identical. It means the two daughter cells have similar genetic composition and as their parents do. Well, we have the mitosis. After the mitosis, we have the meiosis. Occurs in eukaryotic cells and males, and it makes at first the haploid, means in 4 haploids, and then daughter cells that come out form diploid parents, that means 2 in father plus mother. In this process, each has half chromosomes of the parent cells, and that is why it is known as the reductional division. It is reduced to half, and that is how it makes the full. That is why it makes the full. Here is the diagram for your mitosis, mitosis and meiosis is given. Well, children, we have already discussed about the mitosis and meiosis. Here the diagram, it shows that the direct division of the cells into 2 daughter nuclei. Finally, and obviously they are resembling their parents. Here you see in meiosis, the synapses and the crossing over of the chromosomes go, the homologous alignment is there, and then finally to separation of the chromosomes does, and then daughter cells form. And in the daughter cells we get, they are not genetically identical to the parent cell, because you know in meiosis the hereditary character is definitely changed. Alright, now here is the mitosis or somatic body cell division. Here you see the chromosome aligned at the metaphase, and then we have the daughter chromosome separate and to form the 2 daughter nuclei. The daughter nuclei are genetically identical to the parents. So this is how cell division goes on, and I hope you have understood. Please go through your books and look at the pictures nicely. The next day when we come for the lecture, the cell cycle onwards will be discussed. Thank you and have a good day.